Hello everyone. So today we have Sandali from University of Peradeniya. So she is going to talk about an accurate plant pre-microRNA classifier with feature impact analysis. So the stage is yours now, Sandali. So, yep, so just
Thank you. I'm Sandali from Faculty of Engineering, University of Peradeniya, Sri Lanka. Today, my talk is about an accurate plant pre-microRNA classifier with feature impact analysis. I hope you can see three pictures in this slide, uh, and I hope you can identify them. They are different types of cells in your body, a neuron, a skin cell, and a muscle cell. Our bodies have many different cells, but all these cells have the same genes. So, what makes a nerve cell different from a skin cell or a muscle cell? It is because of gene regulation. In other words, turning a gene on or off. MicroRNA comes to the picture at this point. MicroRNA plays an important role in gene regulation. So, what is microRNA? MicroRNAs are these small RNAs that are processed from these longer transcripts that fall back on themselves to make these hairpin structures like this image. This is a pre-microRNA, and this red colored sequence represents a microRNA. In our study, we focused on plant pre-microRNA identification. Uh, Compared with animal microRNAs, plant microRNAs identified 10 years later. In 2002, several groups found plant microRNAs by cloning small RNAs. Since then, different approaches have been employed to identify new microRNAs. Currently, there are four approaches. We can list them as genetic screening, direct cloning after isolating small RNAs, computational strategies, express sequence tags analysis. Genetic screening and direct cloning after isolating small RNAs are experimental methods. Genetic screening is time consuming and very expensive, and also it is difficult to identify microRNAs expressed at lower levels. And talking about direct cloning after isolating small RNAs, it has uh, to overcome the shortcomings of genetic screening, but however, it is still an expensive experimental method. Talking about computational strategies, it is based on genome sequence. There are some computational programs such as MIR scan, MIR align to predict microRNA genes in Arabidopsis, rice, human, and other animals. However, these predicted microRNAs need to be confirmed by experiments such as cloning or Northern blotting. The fourth approach is express sequence tags analysis. This approach is useful for predicting microRNAs in multiple species, especially in species whose genomes are unknown and or poorly understood. However, this method can only identify conserved microRNAs. Most microRNAs are likely non-conserved, so it is impossible to find these genes based on the EST approach. Compared with other mature research areas of gene prediction, predicting and annotating microRNAs still need more work. The four existing approaches have their own advantages and shortcomings, and we can, so that we can combine those approaches and make an efficient way to find microRNAs in plants. So why we did this? There are many microRNAs in plants, but very few of them are identified yet. Plant microRNAs are important in plant development, making stress responsive plants, and also for plant diseases. So studying microRNAs helps to investigate methods to improve crop plants and make medicines for various plant diseases. There are some drawbacks, as I said before, in genetic screening and direct cloning after isolating. Genetic screening is very expensive and time consuming. The cloning method are difficult to find microRNAs expressed at lower levels. So that computational methods have become an efficient alternative in all the ways of for the discovery of novel microRNAs. Okay, the fundamental approach behind the fundamental principle behind our approach is to learn from known microRNAs and identify novel microRNAs. So this is the design methodology. First, we collected data from MIR-based version 14. Then we extracted features of microRNAs under three categories. They are features from micropred, triplet elements, and motif features. After that, the feature selection and the model training were done. Here we selected 4 machine learning classifiers to to build the models, and we evaluate their performances using accuracy, specificity, and sensitivity values. Also, we compared them with past studies, finally obtained the most accurate model to discover plant microRNAs. Uh, this diagram shows a very brief idea about our hierarchy of positive training and testing data set. Here we used a data set generated by the study called Plant miRNA Pred using MIR-based version 14. It is because there are several studies that use the same data set introduced in this study, so that we can compare our models with the past studies. MIR-based version 14 is a primary online repository for microRNA sequences and annotations. There are 2043 known plant pre-microRNAs in the database. They have eliminated the special sequences with complex secondary structures, and then also the important point is the classification performance of a model is highly dependent on the selection of training dataset. So that, and also they have noted that the real pre-microRNAs from the same species and the same microRNA families are highly similar to one another. Therefore, to avoid overfitting, that may occur due to these redundant sequences, they have followed a sample selection method. So after that, 980 pre-microRNAs from 29 species have been selected as the training dataset, and the testing set contains 1,438 plant pre-microRNAs from 8 species. The negative training and testing dataset is also obtained from the previous study. They have used phytosome version 6 to extract the protein coding sequences as the negative dataset. Um, from it used CDS sequences from Arabidopsis thaliana and Glycine max species as proportional to the length distribution of the test, positive training dataset. Following the same sample selection method, 980 pseudo-hairpin sequences selected as the negative training set, and the remaining sequences were taken as the negative testing set. A feature selection method and feature extraction method is a very important step in building a machine learning model. Here we selected 48 features used in the study called MicroPred. This is developed for human pre-microRNA classification. We applied these features to see the impact of them in plant pre-microRNA identification. Those 48 features include thermodynamic and compositional features. Under the triplet elements, we calculated 32 such features considering every 3 adjacent nucleotides. These features were calculated using the predicted secondary structures by a web server called RNA-fold. The next feature set is motifs. Um, they are short range sequences which occur frequently among a set of pre-microRNAs. We used MEME suite version 5.1.0 to discover motifs. We discovered 100 from a positive training dataset and another 100 from negative training dataset. Finally, there are 280 features considered. Uh, we found that there are some highly correlated features among them. So we did a principal component analysis to reduce the dimensions of the data set and obtain 200 principal components. So the idea of principal component analysis is to reduce the dimension of a large data set that still contains most information in the large data set. Here we used 4 machine learning classifiers. They are support vector machine, random forest, multi-layer perceptron classifier, and extra-tree classifier. SVM is a supervised machine learning algorithm that performs classification by finding the hyperplane that differentiates the 2 classes very well. Most of the studies have used SVM as their classifier in pre-microRNA classification. Both the random forest and extra-tree classifier are ensemble learning models fundamentally based on the decision tree algorithm. Random forest models reduce the risk of overfitting by introducing randomness. Extra tree is like random forest in that it builds multiple trees and splits nodes using random subsets of features. But there are 2 key differences in random forest and extra tree. That is, extra tree samples without replacements, and nodes are split on random splits but not best split. MLP classifier, in other words, multi-layer perceptron classifier, relies on an underlying neural network to perform the task of classification. Since there are no studies that used neural network algorithm for plant pre-microRNA identification, we included MLP in our study. Initially, all the classifiers were trained with their default parameters. This table shows the summary of our study. So the first 4 columns show 4 studies that have used the same dataset, SVM classifier, and different feature sets. These values were directly taken from their studies. Uh, the highlighted 4 columns are our studies. You can see that uh, our studies have performed well than the past studies. Uh, some of them have perfect accuracies on several species. For example, the MLP classifier has best accuracy values than the other models for almost all the species except the negative set. And for the negative data set, extra tree classifier and SVM classifier have best accuracy values. The bottom table shows the accuracy, specificity, and sensitivity values for the whole test set. Extra tree classifier and SVM classifier have achieved the best near perfect specificity of 99%, whereas the MLP classifier has got the best sensitivity of 98%. When considering both specificity and sensitivity, random forest classifier has got balanced values of 97% and 96% respectively. Since its overall accuracy is also high, we selected random forest classifier as our final model. And there's a special point that should be highlighted. Even though most of the studies have used SVM classifier and have gained promising results on their studies, in our study, the SVM classifier have less accuracy values than the other models. I think it is because of the feature set we selected. Yeah. Next, move on to the next slide. Here you can see we have tested our models as feature wise. So micropred features have gained 98% for accuracy, specificity, and sensitivity, and motifs around 85, 87 for accuracy, 83% for specificity, and 89% for sensitivity. And comparing the each models, the micropred features have outperformed than the other 2 models, and motif features have low accuracy value than the other models that used micropred features and triplet element features. So, and we can see that when we combine all these features, micropred features, motif features, and triplet element features, we have gained 98 accuracy and 99% specificity and 98% sensitivity. So we can conclude that combining all features can gain higher performance than training the models with motif features, triplet element features. So this that the previous values can also further describe by you seeing these chi-square scores. We calculated the chi-square values of the features and ranked according to the score. The chi-square statistic is commonly used for testing relationships between categorical variables. The features are represented as the symbols used in the almost all the studies that goes under the plant miRNA prediction. There are 33 micropred features and 16 triplet element features and only one motif features among the top 50 features. So these values can more describe from the chi-square test, because the model that use only motif features have low accuracy, and this table also confirmed that the motif features have low impact on the plant miRNA prediction. Uh, yeah. So, as the conclusion, we can conclude that the real and pseudo-pre-microRNAs can be classified with 97% accuracy by including compositional, triplet element, motif, and thermodynamic features with random forest classifier. And although the microped focused on human microRNAs, the features have not negatively affect on the performance, so that the features introduced in microped also can be used to identify the plant microRNAs. Uh, the results have shown that the combination of thermodynamic based and sequence based features have more impact than the motif features. And also, we should talk about the computational time. The computational time to calculate the features in the micropred and the motif extraction took more time than the other features. Therefore, reducing the computational time for feature extraction would be more effective when we integrating the model with a user-friendly software. So, uh, so that it will add a value to your study as well. So, thank you very much. If you have any question, this is time to ask.
Thanks, Sandali. That was a very wonderful presentation. So now I will. Are there any questions for her? Anyone? Okay. So let me start with the first question before anybody else asks. What is pseudo-pre-miRNAs?
They are that they are sequences of nucleotides that we can use them as a negative data set. I mean, when we create a classifier, because it has two classes, we need one data set for pre-microRNAs and another data set for not microRNAs. So here the microRNAs are non-coding RNAs. So that we took coding RNAs, coding sequences as the pseudo uh, sequences,
Oh,
microRNAs.
okay. Cool. And what made you use these, different four different classifiers? Any specific?
Sorry.
You have used four different classifiers, right? Yeah.
Yes.
So what made you choose that? Is there any specific motivation for that, or?
Yes, as I said before, we choose SVM
Mhm.
because almost all the studies uh, that did under the plant miRNA prediction have used SVM, and but they have used different feature sets. So we use that SVM to see the impact of our feature set, because the classifier is same and the data set is same, but our feature set is different, so that we can compare with other studies. And the next one, MLP, is because the neural network based approach is haven't used from in the past studies. That's why we It may be because of the lack of the data. For neural network, we need more data to train the model. So we tried that algorithm to test whether we can further improve the model classification using neural network based one. Yeah.
Cool. Thank you so much. So, any
Thank
questions for her? Okay, we will wait for like, few more minutes.
Okay.
That was a wonderful presentation. You explained everything very
Thank you.
well. Any questions, anyone? I think we have no more questions. So thank you so much, Sandali. That was really
Thank you.
good presentation and all the best.
Thank you. Thank you very much.